Hello, today we're going to be talking about memory, and I'm assuming that you guys all kind of hoped I'd give you some tips and tricks on how to memorize all these facts a little bit better. But unfortunately, today we'll be talking about immunological memory, not neurological memory. And immunological memory is a very long-lasting protective adaptive immune mechanism that's maintained after an infection is successfully eliminated. And this is really critical so that our bodies are able to very quickly and rapidly respond to infections that we might get season after season, such as flu. So today what we're going to be going over are very general principles of immunological memory. And as I mentioned, immunological memory is mediated by the adaptive immune response. So within the adaptive immune response, we have T cells and B cells, and we'll be going over the specifics of memory B cell responses and memory T cell responses as well. So let's start off with some general properties of immunological memory. As I've mentioned, immunological memory is mediated by T and B cells. And after immunological memory is induced after the clearance of a primary infection, secondary responses are mainly attributed to memory lymphocytes. So the lymphocytes that act most powerfully in secondary or third, etc. infections are the ones that are memory lymphocytes. And we're also, towards the end of this, and we're going to cover a little bit on what's called the original antigenic sin. So let's start off with lymphocyte-mediated immunological memory. So immunological memory is one of the most important consequences of adaptive immunity. It's what enables our immune system to respond very rapidly and efficiently against pathogens that are coming in during a secondary infection, or vaccines also enabled us to gain an immunological memory, as a vaccination serves as kind of a pseudo-primary infection. After a primary infection or a vaccination, there's a small group of effector T and B cells that'll then form during that immune response to then persist in the absence of antigen. And what I mean is that these are the cells that are going to form your memory pool once your antigen is cleared. If we look at this example, which is showing a plot of how many cells are within a patient after a smallpox vaccination, you can see that, so this is the time after vaccination on our x-axis, and our y-axis is the relative magnitude. So you can see that after the infection, we have a huge spike in antibody concentration, CD4 cells, and CD8 cells. After a while, after our primary response to the vaccine is cleared, we still maintain high levels of antibodies against smallpox, as well as a large population of CD4 and CD8 memories that are specifically against smallpox, such that after this vaccination, if you were to encounter smallpox, your body would be able to very quickly and rapidly react against the pathogen with the memory pool of CD4, CD8 cells, as well as your antibodies. Now, secondary responses are mainly attributed to memory lymphocytes. So this is primarily due to the difference in the kinetics of responses between naive lymphocytes and memory lymphocytes. So first, we have free antibodies that are floating in our blood at all times from past infections. Free antibodies are able to bind pathogens readily within the blood or within the tissues, and this happens very, very quickly. Then, within a matter of hours or days, our memory lymphocytes are activated to rapidly respond against the invading pathogen. And then, only days or weeks after the preliminary infection or the secondary infection, are more naive lymphocytes activated. So this means that our memory responses are the most active during the secondary response, and it takes a while for our naive lymphocytes to be reactivated. So memory lymphocytes have actually a very curious property in that they actually reduce the activation of naive B and T cells during subsequent encounters with the same antigen. such that passively transferred antibodies to naive recipients inhibit naive B cell responses to the same antigen. This also applies to T cells. So, for example, if you were to get a vaccination against smallpox, as we discussed in a previous slide, if you were to then get infected with smallpox after the vaccination, your memory T and B cell subsets would prevent an additional different response against that smallpox vaccine. Now, why is this important? I'm going to give you a very interesting and important clinical example where this property is used. This property is used to actually prevent the hemolytic disease of the newborn. So if we look at our red blood cells, within the population, you can either be Rh positive or Rh negative. What that means is you can have red blood cells that either have the rhesus factor or Rh on their surface, here depicted by these green dots, or you can be Rh negative, which means that your red blood cells do not have Rh or rhesus factor on their cell surface. Now, if you were to have two people come together and make a child, this child and have the parents be have the male be Rh positive and the mother be Rh negative, the child will actually be Rh positive. This means that the mother will have a protein within her circulation, as there's some sharing of blood, that she will actually generate a response to. So because during the development of her immune response, the mother did not have the elimination of Rh antibodies from her system, the mother will actually gain an adaptive immune response against the Rh factor during the primary pregnancy, which then, were she to be pregnant again, these antibodies would then attack the red blood cells of the fetus, leading to the breakdown of the red blood cells, leading to hemolytic disease of the newborn. Now, given the property that I told you before, that passively transferred antibodies will prevent the induction of an immune response against a secondary infection, you can use this to prevent hemolytic disease of the newborn, such that if there was a woman who was pregnant with a child, if there was a woman who was Rh negative, who was pregnant with a child that is Rh positive, her physician can actually give her Rh-specific antibodies intravenously, so this is passively, and the presence of these antibodies, which is like a pseudo memory response, will actually prevent her from generating her own adaptive immune response against the Rh factor, such that were she to get pregnant again, she wouldn't have an adaptive immune mediated response against the Rh factor in the blood. So using this property of memory response, we can actually prevent hemolytic disease of the newborn, which is incredibly fascinating. Now, another topic I wanted to cover was called original antigenic sin, which is another general property of immunological memory. So what this means is, I want to illustrate this with this following example. Let's say that you have an individual who at a very young age was infected with influenza virus, and this individual generates antibodies against the different types of epitopes on this virus. As you may know, influenza is a very different virus season to season and has different epitopes at different years. So in this example, this individual at a young age generated an adaptive immune response against 4 different influenza epitopes. Now, later on, at 5 years old, or at any given age, but in this example it's 5, if the same individual 3 years later was infected with another virus that had some of the similar epitopes as the first virus, they will actually only generate antibodies against the epitopes that were shared with the original virus. So this is similar as before. So the presence of one adaptive immune response will suppress the other. So here you can see that this individual is actually making more antibodies against epitopes A and C, which they've reacted to previously, and do not react to the new epitopes that are found in the next virus. Now, if we look at the same individual infected with another variant many years later that has various different components of the virus, we can see again that this individual is only responding to the epitopes that are shared with the original virus. Now, this is a property that is in general, for both B and T cells, and it's a very curious thing that is still being actively investigated, but it's something to keep in mind when you're thinking about vaccinations and exposure to pathogens' influence on the memory responses that we have. And now that we've gone through some very general properties and some really cool facts about memory responses in general and how we can use memory responses to cure some diseases such as the hemolytic disease of the newborn, let's jump into general, more specific properties of memory B cell responses. Now we're going to go through the origins of memory B cells. We're going to go through some examples of how memory B cells' responses are more rapid and have higher affinity for antigens relative to naive B cells. And we'll also talk about how memory B cells can actually re-enter germinal centers and gain affinity through additional rounds of somatic hypermutations. But let's start at the start and go figure out where memory B cells come from. So memory B cells can persist in the human body over decades, and they maintain memory for a given antigen without the need for constant antigenic stimulation or proliferation. If you can remember, during a primary immune response, you need to have the presence of the antigen to allow for the proliferation of the adaptive immune cells for which those cells are specific. However, for memory cells, they can actually persist within the body without the antigen being present, which is really important because this allows the cells to be maintained throughout the body and then become activated only when the antigen enters the body again. Now, there's two different types of memory cells and two different origins of memory B cells. The first is memory B cells that are germinal center dependent, and we also have germinal center independent memory B cells. And these are defined based on their origins. So germinal center dependent memory B cells form within the germinal center, as their name suggests. So within the germinal center, during the primary infection, B cells undergo class switch recombination and also gain affinity for their antigen through somatic hypermutation. Now, a subset of these very high affinity B cells, so B cells that have undergone somatic hypermutation such that their B cell receptor or immunoglobulin has a high affinity for their antigen specificity, a subset of these high-affinity B cells within the germinal center will then be selected, and these will differentiate into memory cells. And this process is highly dependent on TFH cells. So this is one class of memory B cells. So again, remember that these are high affinity and class switched, predominantly to IgG, but it depends on the infection. Now, germinal center independent memory B cells actually show no signs of somatic hypermutation. And actually, this is how they were identified. Scientists were looking at memory B cells, and that they found that there was a subclass of them that did not undergo somatic hypermutation, which implies that they did not enter the germinal center. Now, these responses, the germinal center independent memory B cells develop very, very early in an adaptive immune response before B cells actually enter the germinal center to undergo somatic hypermutation. And these form without the dependency on T cell help. So, just so we can remember, we have two different types of memory B cells that have different origins. The first, we have our germinal center dependent memory B cells. So these are the cells that have very, very high affinity antibodies, and they are class switched, and they develop from the germinal center interaction that allows them to gain high affinity. We also have germinal center independent memory B cells that have not class switched and do not have somatic hypermutation. So these cells have a lower affinity relative to the germinal center dependent memory B cells. So we have memory B cells that are formed before and after the germinal center reaction, such that we have a pool of memory B cells that are both high affinity for specific epitopes, but we also have a subset of memory B cells that are specific for the antigen, but not so high affinities so that they could, this allows for the germinal center independent group of B cells to potentially interact with epitopes of pathogens that have not evolved over the years. Now that we've talked about the origin, let's talk about where these memory B cells hang out. So first, if we have a pathogen that's encountered in the lymph node, so again, just as a reminder, pathogens are presented in lymph nodes if they're encountered in the tissue. So pathogens that infect a tissue will then be presented in lymph nodes. So lymph node specific pathogens will interact with their B cells within the lymph node, obviously. And memory B cells, some of them will stay within this follicular mantle in the secondary lymphoid follicle, and some will actually leave this original lymph node where they were generated and enter the blood. So they'll flow throughout the blood to all different lymph nodes. So when a pathogen is encountered in the tissue, most of the antigen-presenting cells that will present the pathogen epitopes will migrate to the closest lymph node, but you can have a pathogen invade a tissue all over the body. So if you were to form a memory B cell response, say in your neck, you would want to make sure that these B cells that could then act against a secondary infection are spread throughout the body. So we do maintain some memory B cells within this follicular mantle. However, some will leave and circulate through the body. Just as a reminder, as these cells are formed within the germinal center, the majority of these memory B cells that stay within the lymph node are germinal center dependent B cells. Now, if a pathogen enters the host through the blood and such is encountered in the spleen, then the B cells will actually congregate in splenic marginal zones, seen here. And this is very parallel to your lymph nodes, and they'll predominantly stay within the spleen because they don't need to recirculate, as most blood-borne pathogens will be encountered within the spleen. So just to summarize, we talked about pathogens that are tissue-specific that'll be encountered in the lymph nodes. So these are memory B cells. Some will stay within the specific lymph node in which they were developed, and then others will leave and circulate through the body. And then pathogens that are blood-borne that are encountered in the spleen, the memory B cells that are formed there will stay there within the marginal zone. Now, memory B cell responses are much more rapid relative to naive B cell responses, but they also have a very high affinity for their antigen if they are germinal center dependent B cells, which is the majority of the memory B cell response. Here I want to go over this chart, which you can find in your Janeway textbook, to talk about the different properties of memory versus naive B cells. Here, this is just comparing different properties of B cells from an unimmunized donor and an immunized donor. So the unimmunized donor is a representation of a primary response. So these are naive B cell responses, and our immunized donor is a representation of memory responses. So if we look at our unimmunized donor, there's a fairly low frequency of antigen-specific B cells, and we mostly have IgM antibodies being produced, again, because these are cells that have not class switched. And they have a very low affinity to their antigen, and they have very low evidence of somatic hypermutation. Now, on the other hand, if you look at someone who's already been immunized to this pathogen, they have a lower frequency of these antigen-specific B cells, but they are of much higher affinity, and they are predominantly class switched, so they're IgG or IgA specific. And then we have a very high affinity of the antibody as they've undergone somatic hypermutation, and as I said, they have very high evidence of somatic hypermutation.